Khateeb series in Human Anatomy and Physiology. Chapter 2, Cell Structure and Function. Episode 2. Cell junctions and their functions. In the world of cell biology, cells within tissues do not function in isolation. They communicate, adhere, and form barriers to maintain the integrity of organs. Cell junctions play an essential role in maintaining the structural integrity and functionality of tissues. Among the various types of junctions in the cell, tight junctions, desmosomes, and gap junctions stand out as key players in ensuring the proper functioning of cellular networks. These junctions help regulate the movement of substances, provide structural support, and enable communication and electrical coupling between cells. Today, we will discuss each of these junctions in detail, exploring their structure, function, and the roles they play in maintaining tissue health and homeostasis. The first and a common type of cell junction is the tight junction, also known as the zonula occludens. These junctions are impermeable barriers found at the apical region of epithelial cells, with their major function being to form a seal between these cells. Let's magnify the tight junction indicated in this image to understand its structure. Structurally, tight junctions consist of a complex network of transmembrane proteins such as claudins, occludens, and junctional adhesion molecules, JAMs. These proteins work together to seal cell membranes of adjacent cells. There are also cytoplasmic proteins like zonula occludens 1 and zonula occludens 2. That link tight junctions transmembrane protein to the actin cytoskeleton, contributing to the structural integrity of the junction. In addition to sealing, tight junctions serve the epithelial tissue with many functions. Yet, their primary function is to maintain selective permeability, preventing the passage of molecules and ions between adjacent cells. This is particularly important in tissues like the intestines and the blood-brain barrier, where tightly controlled transport is crucial for function and protection. As shown in this image, tight junctions also act as a defense mechanism, restricting pathogen entry into epithelial layers, which are especially important in the intestines and the blood-brain barrier. However, loss of tight junction integrity, for example in the gut, leads to leaky gut syndrome such as those observed in celiac disease. The second and common type of junctions between the cells is the desmosomes. Desmosomes are specialized cell-to-cell junctions that provide mechanical strength to tissues by anchoring cells together. These junctions are crucial in maintaining tissue integrity which are subjected to high mechanical stress, as in the skin, heart, and uterus. The structure of desmosomes consists mainly of transmembrane proteins called cadherins, which extend across the plasma membranes of the cells and interact with their counterparts on neighboring cells. Examples of cadherins are desmogleins and desmocollins proteins. These cadherins are linked to the intermediate filaments of the cytoskeleton via intracellular plaque proteins, such as desmoplakin and plakoglobin. This connection forms a strong adhesive bond between cells, providing structural stability to tissues. The last type of junction is the gap junction. This type of junction are unique intercellular permeable connections that facilitate direct communication between adjacent cells. Therefore, gap junctions are very common in tissue that are metabolically active or require rapid communication such as the heart, smooth muscles, skin, liver, neurons, and cornea of the eye. Based on this, we can conclude that the structure of gap junction is mainly made of special channels called connexins, also known as hemichannels, that cross the plasma membrane of each cell. As shown in the image, each connexins is made of 6 individual proteins known as connections. When 2 connexins from 2 neighboring cells align, they form a complete gap junction channel as shown in this image. These channels allow the easier communication between the cells by exchanging ions, metabolites, and small signaling molecules. This makes gap junctions play several key roles in cellular function. For example, they allow for synchronized electrical impulses in tissues such as cardiac and smooth muscles, enabling coordinated syncytium contractions as one unit. In the same manner, in neurons, gap junctions facilitate rapid electrical signal transmission, essential for communication between nerve cells. Additionally, gap junctions play crucial roles in embryonic development by facilitating coordinated cell growth and differentiation, as well as in wound healing, where they enable rapid cell communication to regulate the repair process.